Welcome to all viewers and well-wishers of the world. The purpose of my lecture is to learn the knowledge from the word and to return the knowledge to the word. Motto of my life is learn, earn and return. I have learned a lot, earned a lot. Now this is the time to return a lot to the word from where I have got the knowledge. The topic of today's discussion is bacterial classification. It's a very important topic and students of microbiology will definitely have a lot of help from him. Classification of bacteria, simply we divide the kingdom, phylum, class, order, family, genus, the first name of the bacteria, species, the second name of the bacteria, or the identifier who have identified the bacteria. So simply we can just say that if you remember this sentence, King Philip came over for good spaghetti. So you can remember it like that. Now the general concept, classification is orderly arrangement of the bacteria into groups. Bacteria are classified and identified to distinguish the bacteria one organism from the another, and to group the similar organism in a one group. Level of this type of a classification is the species level. Species group of the similar organism within a genus is called species. There are many bases of classification, some of which I am going to give it here and to describe it here. Basis of the classification of the bacteria, one can classify by the different way, but the basis which I am going to describe with you people is the number one is the Gram-Stain reaction. It's the shape of the bacteria, the arrangement of the cell of bacteria, motility, the capsule of the bacteria, spore formation, oxygen requirement, host-parasite relationship of the bacteria, then cultural characteristic of the bacteria, biochemical reaction of the bacteria, antigenic structure or serology of the bacteria, genetic composition of the bacteria, phage typing, flow cytometry, and disease they cause. These are the different bases of classification of the bacteria. So the classification first, the Gram-Stain reaction, as people know and the students know, there is Gram reaction. On the basis of that, the bacteria are classified. So these are Gram positive or Gram negative. The Gram positive are further divided into Gram positive cocci or the Gram positive bacilli. Gram positive bacilli are further divided subdivided into non-spore forming, and these are of two types, like non-filamentous Corynebacterium and the filamentous Actinomycetes. Or the gram-positive bacilli can be the spore forming, which are of two types, that is aerobic, the Bacillus anthracis, and the anaerobic, the Clostridia, which causes tetanus. Now the gram-positive bacteria, for the convenience one can see the gram-positive cocci, these are Staphylococcus and Streptococci. Then gram-positive bacilli, these are the Bacillus anthracis and anthrax bacteria. And these are again, the gram-positive bacilli are divided into spore forming and non-spore forming. The spore forming are again subdivided into aerobic, which are the Bacillus anthracis, and or anaerobic Clostridium bacteria. Now one can see the Staphylococcus bacteria and the Bacillus anthracis, which are like this, then Streptococcus pyogenes, which looks like this, and the Listeria bacteria. These are the bacilli and these are the cocci. So one can differentiate very easily that these are the rod shape and these are the cocci or the berry shape or the round in shape. Similarly, the gram-positive rods, these are the spore-forming. These can be aerobic and anaerobic. As one can see, the spore inside the bacteria. So these are called spore-forming bacteria, and that is seen in Bacillus anthracis. And the other are non-spore forming. These are the Corynebacterium, and these are the Actinomyces israeli. These are, one can see, that is the filamentous, and these are the non-filamentous. Then gram-negative bacteria include gram-negative cocci. These include gonococcus and meningococcus, which cause meningitis. Gram-negative bacilli, these are further classified as Enterobacteriaceae and other gram-negative bacteria, which are very important for causing the different diseases. So the gram-negative bacteria, these are divided into bacilli and the cocci. Cocci are the meningococcus and the gonococcus, and the bacilli are further divided for the convenience purpose, Enterobacteriaceae and the other gram-negative bacilli, which does not come into the Enterobacteriaceae. Or then other classification for the convenience is the respiratory, zoonotic, or enteric. Respiratory, which cause the respiratory diseases; zoonotic, which are the diseases which are coming to the human being through the animals; and the enteric, which are involving the intestine of the human being. So these are called. Now the very important is the Enterobacteriaceae, that includes 13 bacteria, and this is most of the time being asked in the Viva, so one should remember it by heart, and very easy to remember. I will let you know in the next slide. These are Morganella species, Yersinia species, Salmonella species, Shigella species, Serratia species, Protea species, Providentia species, Citrobacter species, Daphnia. It is controversial in some literature. Escherichia species, and Enterobacteriaceae species, Edwardsiella, and Klebsiella species. Now, these 13 bacteria are very simple to remember by this way, which I have made, that is Enterobacteriaceae. If you see any policeman fatty, so that is SSP of the police. You can remember like this, my SS, because he's a fat, so instead of SSP, he has 3S, and P, he is also having not 1P, 2P, and he have got the large size of cheeks, so this is having one extra E. So you can remember like this, my SSP cheek: Morganella, Yersinia, Salmonella, Shigella, Serratia, Proteus, Providentia, Citrobacter species, Daphnia, Escherichia species, Enterobacter, Edwardsiella, and Klebsiella. That all makes my SSP cheek. So one can remember like this and can answer during the exam. So one can see the Enterobacteriaceae bacteria. As you know, these are all gram-negative and bacilli. So that is Citrobacter, that is Salmonella, this is Shigella, and this is E. coli. And other gram-negative bacilli which does not fall into Enterobacteriaceae, these are the Campylobacter, Helicobacter, Acinetobacter, Vibrio, Pseudomonas, Bordetella, Francisella, brucellosis, Haemophilus species, Gardnerella vaginalis, Legionella, and Pastorella. Here you have to differentiate between Enterobacteriaceae and the other gram-negative bacteria, and there is a very slight difference. As one can see in this photograph, there is a very slight difference, but one can appreciate what is the difference between the two. So classification depending upon the bacterial shape. Bacteria are of 9 shapes, so according to that it have got the 9 different kind of shapes and classification depending upon the different shape. Cocci, these are round, bead shape like Streptococcus and Pneumococcus. bacilli, as I have told you, the rod shape, these are the E. coli, salmonella and proteus. Vibrios, which are slightly curved or comma shaped, not straight rods as the bacilli, so these are the Vibrio cholera causing the cholera. Then spirelia, which are spring shaped, regularly coiled rigid spiral or helical forms, that is the Spiralium minus. These are the bacteria which are having spirelia shape and spring shape. One can see that the cocci, bacilli, Vibrio, as you can see, the comma shape, and here we can see the spiral. These are the Spiralia bacteria. Then the classification on the shape continued. Spirochetes, these are the loose spiral forms, tightly coiled, as in case of the Treponema and Borrelia. Actinomyces, these are the branching filamentous bacteria, resemble to the filament of the fungi. But these are basically the prokaryotic cells, and the fungi are the eukaryotic cells, but they behave and they appear like the filamented. Then wall-less or mycoplasma. These lack the cell wall, bounded by a single triple layered cell membrane and not cell wall, so these are called wall-less. Pleomorphic, these bacteria have many shapes or variable shape like coccobacilli, so neither they are cocci nor they are bacilli, so because of that shape these are called pleomorphic. Then sometimes the rare form of the bacteria is seen like protoplast, spheroplast and L forms. These are basically the defective or absent cell wall during development of the bacteria or during the certain treatment with a different antibiotic. The bacteria get shape, change their shapes, and they look like the protoplast or spheroplast or the L form. So here you can see the spirelia, and there the filamentous shape bacteria of actinomyces. Other form of the bacteria, one can see the pleomorphic, different form, filamentous like filaments, star shape like star, box shaped bacteria and appendaged bacteria, and these are the sheath bacteria. So these are all the different form and the rare form. And also you can see the gram-positive bacteria, they can change to the protoplast, and gram-negative bacteria, they can change to the spheroplast. And similarly, these are the cells which can change into the L form, that is during process or during development or during the treatment with a certain antibiotic, they can get this new shape and change because of their lost sometimes the cell wall or the altered cell membrane, they have got this shape. Then classification of the bacterial arrangement, how the bacteria are arranged. So these are the cocci. The cocci do arrange into pairs. So when they are in pairs, these are called diplococci. When they are in chains, these are called streptococci. When they are in clusters or grape-like, when they are called staphylococci. Tetrads, when they are in shape of 4 bacteria. Then cubical packets, when the bacteria lie over one another and give this kind of shape. Similarly, in case of the bacilli, diplobacilli, when 2 bacilli join together. Streptobacilli, when these bacilli are in chain. Coccobacilli, some are of the cocci shape and the other are of bacilli shape. Then branching chains like the filamentous. Palisade arrangement, that is the hedge one can see outside your garden. So the palisade arrangement of the wood that just make the palisade. So these are like that. Massed together, they just heap over one another, and cuneiform or the Chinese letter bacterial shape. So that is the characteristic of the different bacteria to have the different shape. Here one can see the cocci, single, in chain, in pairs, the diplococci, and here you can see the tetrads, there are 4, and these are the sarcina, these are the 8 bacteria on one another, and then there is a grape-like, just a bunch of the grape, so that is called streptococci. Similarly, in case of the bacilli, one can see the single bacilli, the diplobacilli, the 2 bacilli joined together, then the chain of that, and then the different form of the different bacteria that, as I have told you already, that just get the shape. Then classification on the basis of the motility, and one should remember this thing, the basis of the motility is the flagella. So flagella is just like the legs of the human being. By flagella, bacteria can move to anywhere. So bacteria which contain flagella are motile. So for the convenience, these are called flagellated. It means they have got the flagella and they can move. Bacteria which contain no flagella are non-motile, so-called non-flagellated. Examples are the Salmonella versus Shigella. Salmonella have got the flagella so that it can move. Similarly, the E. coli versus Klebsiella. E. coli do have the flagella, so it can move very rapidly, and Klebsiella does not have any flagella, so it is immotile. Here one can see the spherical shaped bacteria, the cocci, streptococci. These are how one can differentiate. These are non-flagellated. And rod-shaped bacteria, bacilli of the Salmonella, one can see the flagella which is attached to one pole of the bacteria. By virtue of that, they are able to move from one place to another place. Similarly, flagellated and non-flagellated, as one can see and appreciate the flagella of the Salmonella bacteria and Shigella, which have got the no flagella, only it have got certain pilus, as you can see the fibers which are coming out from the surface of the body. These are not the flagella, these are just the pili. Now, classification on the basis of the capsule. Some bacteria have an envelope around the cell wall which is called capsule. So these are called capsulated bacteria, as they do have the capsule. Examples are the Streptococcus pneumonia, Haemophilus influenzae, and Meningococcus. These are all the capsulated. These having no capsule are called non-capsulated bacteria. These are examples are Staphylococcus, Streptococcus, and E. coli. They do not have any capsule. So one can appreciate the capsule here that is around the bacteria cell wall, and here there are no capsule. So one can easily see the capsule around the bacteria. So these are called capsulated bacteria.